Okay, hi everyone. We'll start now. So this is the fifth problem solving session of the course Biochemistry. The course instructor is Professor Swagata Dasgupta. You must have seen her lecture videos by now. So I'm Shrija Ghosh. I'm PMRF at IIT Kanpur, and I'll be taking the doubt clearing session for the fifth week. The whole lecture was about enzyme inhibitors and then different enzyme mechanisms. So we'll discuss the questions related to those topics, and feel free to stop me and ask at any point you want. You can type your doubts in the chat box also if you don't want to unmute, otherwise you can ask. Okay, so we'll start. Just a second. Yeah. So starting with the first question. Which amongst the following can act as choline esterase inhibitor? Option A choline, option B insecticides, option C phenyl benzoate, and option D ethyl acetate. So which amongst the following can act as choline esterase inhibitor? Something that can inhibit the enzyme choline esterase. Anybody knows the answer? Anybody wants to guess the answer? Okay. Okay, Manisha has answered option B, insecticides. Very good. That is the correct answer. Insecticides. So, choline esterase is an enzyme that basically can ca- uh, can use acetylcholine as substrate, and it can break it into acetic acid and choline. So acetylcholines are neurotransmitters. Okay, so for transfer of any signal in our body, these small molecules, these are neurotransmitters, they basically carry the information and transfer from one neuron to another. Okay, so for that transfer, these acetylcholine esterases are an important enzyme. Okay, now what happens? So you can see here the diagram. So here this is the acetylcholine, so, and this is the enzyme. So the enzyme has the catalytic pocket, and you can see the substrate fits in that catalytic pocket, and the catalysis occurs where the substrate gets converted into the products. Okay, now what happens when we have insecticides and pesticides? Most of the insecticides and pesticides have phosphate groups present in it. Okay, so in the presence of phosphate, what happens uh, in this pocket, the esteric side that you are seeing, the serine, the OH group of serine, it can form conjugate with the phosphate groups of the insecticides and pesticides, and thus the insecticide molecules that are there, they can block the enzyme active site and prevent the binding of the substrate to the enzyme. Okay, so if you have seen any news about usage of insecticides causing a lot of health disorders and mental disorders, those are actually because of such insecticides that can block these important enzymes in our body that can actually affect the neurotransmission in our uh, body or in our system. Okay, so that's why the usage of these insecticides and pesticides are harmful. So so insecticides are the inhibitor of our cholinesterase enzymes. Okay, so moving to the next question. So just a second. moving to the next question, identify the correct statements regarding the allosteric regulation in enzymes. Now statements are, first statement is, when a small molecule acts as an effector or regulator to activate or inactivate the action of an enzyme, it is said to be under allosteric control. Second statement is, the small molecule generally binds close to the active site of the enzyme. Third statement is, regulation generally arises from the conformational change in the enzyme when the ligand is bound to it. And the fourth statement is, hemoglobin and myoglobin show equal extents of allosteric effect in the presence of oxygen species. So now you have to identify the correct statements amongst these four statements. Okay. So I would request all of you to go through the statements once and try to find the correct statements. Then we will discuss the correct answer. What could be the correct statement among these? Anybody? So let's start with the first statement. When a small molecule acts as an effector or regulator to activate or inactivate the action of an enzyme, it is said to be under allosteric control. Is it true or is it false? If we have a small molecule that can actually activate or inactivate an enzyme, the enzyme, yes, Neeta is saying it is true. Yes, correct. This statement is very true. So if we have a small molecule that can regulate the enzyme mechanism, so if it can activate the enzyme or inactivate the enzyme, in that case the enzyme is said to be under allosteric control. The such molecules that bind to the enzyme and regulate it, those are called allosteric molecules, and they actually bind to an allosteric site of the enzyme. Okay. Okay, Manisha has answered. Option C is the correct one. Okay, so the second statement, the small molecule generally binds close to the active site of the enzyme. That is a wrong statement, because if a molecule will bind close to the active site of the enzyme, it will kind of inhibit the enzyme. Okay, it cannot regulate the enzyme. It will it can permanently actually inhibit the enzyme. Okay, these allosteric sites are away from the active site. Okay, and they actually modify the binding with the substrate. So either it can enhance the binding of the substrate or it can reduce the binding of the substrate. Now third statement, regulation generally arises from the conformational change in the enzyme when the ligand is bound to it. That is again correct, because as I told you, once these enzymes are once these molecules are bound to the enzyme, they actually regulate the binding of the substrate to that enzyme. Okay, and fourth is, hemoglobin and myoglobin show equal extents of allosteric effect. So this is a wrong statement, because hemoglobin shows allosteric effect but myoglobin does not. So hemoglobin is a 4 subunit protein, so it actually shows allosteric effect with oxygen, a small molecule, but myoglobin does not. So option C is the correct answer. First and third statements are correct. Okay, so Manisha, you were right. So here you can see a diagrammatic representation of allosteric regulation. So this is inhibition and this is activation. So you see, this is the active site of the enzyme, and the allosteric site is away from the active site. Okay, it is not near the active site, it is away from the active site. And once that small molecule binds to that allosteric site, so in case of inhibition, what is happening? Once the small molecule is binding to the allosteric site, you will see there will be a conformational change in the active site that will prevent the binding of the substrate to the enzyme. Okay. In other case, in case of activation, what happens? You have the small molecule that binds to the allosteric site, and that small molecule, once it binds to the allosteric site, it changes the conformation of the active site such that now the substrate can actually fit in the catalytic pocket. Okay, so in first case, the small molecule binding is preventing the substrate from binding, that is, it is inhibiting the enzyme to do its activity. Whereas in the second case, the small molecule is actually enhancing the substrate binding by making the conformation of active site such that the substrate can fit in the catalytic pocket. Okay, so this is the allosteric effect of enzyme. So enzyme is under allosteric control or allosteric regulation, as you can say, and a small molecule is actually regulating the enzyme's action. So either it is inhibiting, that is slowing down the enzyme, or it is activating, or you can say speeding up the process of the catalysis. Okay, so we talked about hemoglobin and myoglobin. So both hemoglobin and myoglobin can actually bind to oxygen. Okay, so both hemoglobin and myoglobin can bind to oxygen, but what is the difference? The difference is hemoglobin is actually a 4 subunit protein. So in the last class also we had discussed, so hemoglobin is, you have 2 alpha and 2 beta subunits. Okay, now what happens? Oxygen, which is a small molecule, it can bind to hemoglobin under allosteric conditions. So once oxygen binds to the first alpha subunit, so let's say this is alpha 1 and this is alpha 2. Okay, so once oxygen binds to this first alpha subunit, it kind of facilitates the binding of other oxygen molecules to the other subunits of hemoglobin. Okay, so basically once alpha 1 binds to oxygen, it will facilitate the binding of oxygen to beta 1, then beta 2, alpha 2, and like so. This is the allosteric effect of hemoglobin. So what is the molecule, small molecule that is binding? That is oxygen. It binds to a allosteric site of the hemoglobin. Once oxygen is bound to hemoglobin, it can perform its catalytic function. Okay, this is clear? Okay, so the first statement, that whenever a small molecule regulates in order to activate or inactivate an enzyme, the enzyme is said to be under allosteric control. That was true. And the third statement, that the regulation arises from the conformational changes. So how the conformational change is happening, I've shown it to you. So either in case of inhibition, the conformation is changing such that the substrate cannot bind. In case of activation, the conformation is changing in such a way so that the substrate can bind to the enzyme. Okay, the second statement, that was small molecule generally binds to close to the active site of the enzyme, that is not true. That you can see here in this diagrammatic representation, the allosteric site is away from the active site. Okay, and then I have told you about hemoglobin and myoglobin. So this is clear. Then we will move to the next question. Question number 3. The above reaction is catalyzed by succinate dehydrogenase. Okay, so here you can see the reaction happening. So this, what is this? This is succinate or succinic acid. Succinate is getting converted into fumarate by succinate dehydrogenase. Okay, what will be the product if succinate dehydrogenase was to react with the molecule X? Now, instead of this succinate, if this molecule was there, what would be the product? Anybody? So if succinate dehydrogenase can convert succinate into fumarate, the structures have been given to you. What would be the product if succinate dehydrogenase has to bind with X, the molecule X, molecule shown here as X? Anybody wants to answer? It's not very difficult. Think of it. So the answer to this question is option D. That the reaction will not occur. Now, why the reaction will not occur? If you remember, in the previous class also I have told you, the enzyme is very specific to its substrate. Okay, so I've told you about lock and key mechanism or induced fit mechanism, where the substrate molecule fits into the catalytic pocket of the enzyme. Okay, so whatever the shape of the catalytic pocket will be, the substrate will fit into that pocket very easily. Okay, now if an enzyme is made such that succinate can fit into that pocket, no other molecule will be able to fit in that pocket and do the same catalytic reaction. Okay, even if the molecule is similar to the structure of succinate. So here the molecule X has a structure similar to that of succinate. Still, the catalysis won't happen because those active residues, those are responsible for the catalytic action, like particular amino acids that are going to do the catalytic reaction, those amino acids won't get those particular groups or particular conformation from which they can perform the catalysis. Okay, so do you understand? So every enzyme is specific towards its substrate. So if you give a enzyme some other substrate which is mimicking to the structure, like if I if I try to do the catalysis with a molecule which is mimicking the substrate structure, even then the enzyme won't be able to catalyze the reaction. The enzyme will get, even if the molecule fits in the catalytic pocket, the enzyme will get inhibited and the molecule will stay in the catalytic pocket. The conversion of substrate into product and then release of product from the enzyme won't happen. Okay. Okay. So moving to the next question, question number 4. In the given diagram, the enzyme inhibitor interaction is shown. So E is enzyme and I is inhibitor. Among the paths A and B, indicate probable type of interaction in each path. The respective shapes are indicative of the activity of each species. So here you can see two paths are shown. One where the enzyme is binding to the inhibitor and then later the inhibitor and the enzyme are getting released, like inhibitor is getting released from the enzyme. In the second case, the inhibitor is binding to the enzyme, but when the inhibitor is released, the enzyme shape is changed. Now the shape represents the activity. That means the enzyme's activity has now changed. Okay, so initially the enzyme is represented inside a square. Okay, so if they have represented the square at the end of the process also, that means the activity is same. Otherwise, the activity has changed. As you can see, in case of the B path, the activity of enzyme has changed. Now we have to tell what is the type of interaction between the enzyme and inhibitor that is there during the path A and path B. Okay. So Neeta has answered option D. So path A has non-covalent interaction and path B has covalent interaction. Okay, that is the correct answer. So why does it have a non-covalent interaction in first case and covalent in second case? Would you like to explain? So if you see in the second path, we can see that the activity has changed. That means the bond that was forming between the inhibitor and the enzyme was strong enough that when it was broken, it created a change in the enzyme itself. Okay. Yes, yes. So inhibitor and enzyme are separated after the reaction and bond is broken down. Yes, exactly what I was explaining, Neeta. Very good. So when in case of the path A, what is happening? The enzyme and inhibitor are interacting, but there is no breakage of bond. So when those two molecules are interacting together, when they are separated, the activity remains the same for both of the molecules. But in second case, what is happening? The activity of the enzyme is changing. The change in activity actually shows that there was some covalent interaction that was broken, and due to bond breakage, there has been a change in the activity. Okay, so the first path is non-covalent interaction. The second path shows a covalent interaction between the enzyme and inhibitor. Okay. So second case could be called as irreversible inhibitor. Okay. So in this case, in the first case, once the inhibitor is released, the enzyme is retaining its activity. So that is reversible. Okay. The inhibition is reversible. When the inhibitor is bound to the enzyme is inhibited. When the inhibitor is released from the enzyme is again active. But in second case, when the inhibitor is bound to the enzyme is inhibited. Once the inhibitor is released from the enzyme, even then the enzyme's activity has been hampered. That means the inhibition that has happened was irreversible. You cannot go back to its active state now. Okay, so in case of covalent interaction with the inhibitor, mostly the inhibition is irreversible type. That is, you cannot go back to the active state of the enzyme. Okay. In case of non-covalent interactions, the inhibition is reversible type. Okay. I hope this is clear. Okay, so moving to the next question, question number 5. The graph represents reaction rate versus substrate concentration plots for an uninhibited enzyme and same enzyme in the presence of 3 different inhibitors A, B and C. Which statement about this data is true? Okay, so here we have a graph where the black line is showing the normal enzyme activity. So it is the rate versus the substrate concentration, classic Michaelis-Menten graph. And then they have shown the reaction rate versus the substrate concentration in presence of 3 different enzymes: enzyme A, enzyme B and enzyme C. Oh, sorry, 3 different inhibitors. I am sorry, 3 different inhibitors. Inhibitor A, B and C. So now you have to say the type of inhibition that is happening, or the type of inhibitor that is there in A, B and C. Okay. So anybody wants to answer? So option A says inhibitor A is non-competitive, B is uncompetitive, and C is competitive. B says A is competitive, B is uncompetitive, and C is non-competitive. C says A is uncompetitive, B is non-competitive, and C is competitive. And D says A is competitive, B is non-competitive, and C is uncompetitive. So what is the correct answer? Anybody knows the correct answer? The correct answer is option D, where A is competitive, B is non-competitive, and C is uncompetitive. Now we will see why. Now we will see one by one each of these three inhibitors. Okay. So here in case of competitive inhibition, what is happening? The inhibitor is binding to the enzyme. This gray shaded thing is enzyme. So the inhibitor is binding to the enzyme at the same site where the substrate binds to the enzyme. Okay. So there is a competition between the substrate and inhibitor which will bind to the enzyme first. Okay. So this is clear. So what is happening in competitive inhibition? There is one catalytic site and both substrate and enzyme are binding to that same site. So there is a competition. So if substrate is higher, if substrate concentration is higher than the enzyme concentration, what will happen? This substrate can bind to the enzyme. It can actually replace the basically it will win the competition with the inhibitor. If the inhibitor concentration is more, inhibitor will win the competition and it will bind to the enzyme, rendering it to be inactive. Okay. So in case of competitive inhibition, you can see either substrate can bind to the enzyme or inhibitor can bind to the enzyme. So the total enzyme concentration will remain same. Total free enzyme concentration will remain same. Okay. Now, in case of non-competitive inhibition, what is happening? Substrate can bind to the free enzyme and substrate can bind to the enzyme inhibitor complex also, and inhibitor can bind to the enzyme substrate complex also. So anything can bind to anything at any point. So now we have the enzyme here. Substrate will bind to the enzyme, sit in its catalytic pocket. Inhibitor can bind at this stage to that enzyme substrate complex to form a ternary complex. Okay. In the second case, what can happen? Inhibitor can bind to the free enzyme, and then after that also substrate can bind to the enzyme, but it get converted into product. So product conversion will only happen when there is only enzyme substrate complex present. The substrate will get catalyzed and get converted into product. This is clear. So non-competitive inhibition, substrate can inhibitor can bind to the enzyme substrate complex as well as the free enzyme. Now uncompetitive inhibition, in this case the inhibitor cannot bind to the free enzyme, it can only bind to the enzyme substrate complex. Okay, so here's the difference between non-competitive and uncompetitive inhibition. This part is not there. Okay, so that means inhibitor cannot bind to the free enzyme, inhibitor can only bind after the substrate has bound. So it's a sequential process. So first substrate will bind to the enzyme and then inhibitor will bind to the enzyme substrate complex. So it's a sequential process, whereas in this case anything could happen at any point, like inhibitor can first bind to the enzyme and then substrate, or inhibitor could bind to the enzyme substrate complex directly. Okay, but in this case it's a sequential process. Okay, so this is clear what is competitive, non-competitive and uncompetitive inhibition is. Now now I'll explain you why this graph is representing competitive inhibition, this graph is representing non-competitive, and this is uncompetitive. So if you see here in the competitive inhibition, you can see the total enzyme concentration remains same. The enzyme is there, either the substrate will bind to the enzyme or the inhibitor will bind to the enzyme. In a case where I'll keep the inhibitor concentration constant and keep increasing the substrate concentration, we'll reach a point where the enzyme will reach its maximum velocity, that is Vmax. So if you remember, so if you see this graph here, so if you see this graph here, so this point, the maximum point here in the normal enzyme is showing what? Vmax. Okay. So in case of competitive inhibition, the starting will be slow. Since there is inhibition, the velocity will be slower than the initial velocity. So this is the initial velocity in case of normal enzyme, and this is the initial velocity in case of inhibitor A. So initial velocity is lower because the rate of because there is inhibition, the rate of catalysis will be slower. But eventually, when you keep on increasing the substrate concentration, eventually the enzyme will reach its maximum velocity. So there will be no change in Vmax, but Km will increase. Why Km is increasing? Because now you see, so this is Vmax, so somewhere here we will have the Vmax, half Vmax, and I have told you at half Vmax the substrate concentration is equivalent to what? Substrate concentration is equivalent to your Km. Okay. So this is the Km for normal enzyme. This is the Km for the inhibited enzyme, which is inhibited by the inhibitor A. So A is a competitive inhibition because the Vmax is not changing and the Km is increasing. Okay. So when you plot a Lineweaver work plot for this competitive inhibition, you will find the graph to be like this, where so this y-intercept is showing what? 1 upon Vmax. Okay, so if Vmax is not changing, its reciprocal will also not change, and x-intercept is showing minus 1 upon Km. So that is increasing. So minus 1 upon Km will decrease, right? Reciprocal will decrease. So this is the inhibited one and this is the uninhibited one. Okay. Yeah. So in case of competitive inhibition, this is clear why Vmax is not changing and Km is increasing. Now we will see the second case, the B. Just a second. So now we will see what is happening in case of inhibitor B. So in case of inhibitor B, we said that there is non-competitive inhibition. So if you see, the initial velocity has reduced and the Vmax has reduced, but the Km, so this is the Vmax for this, and somewhere here we will have the half of Vmax. Okay, somewhere here we will have the half of Vmax. You will see the Km for both normal enzyme, uninhibited enzyme, and the enzyme that is inhibited by the inhibitor B, the Km is same. Okay, in case of non-competitive inhibitor, there is no change in Km and the Vmax decreases. Okay, there is no change in Km and the Vmax decreases. Okay, so in case of non-competitive inhibitor, you will see there is no change in the Km. Why there is no change in the Km? Because what is Km? Km basically determines the binding efficiency of the substrate to the enzyme. Now, if you see, there is no difference in the binding efficiency of the enzyme, binding efficiency of the substrate, because substrate can bind to the free enzyme as well as in presence of inhibitor also. So there is no problem in binding. There is a problem in the catalysis. Okay, so the catalysis is slow. Okay, Kcat is decreasing. Why Kcat is decreasing? Because the Vmax is decreasing. Now all the substrate, this ESI, all the inhibitor enzyme substrate inhibitor complex, this ternary complex will not get converted into enzyme and product. So only enzyme substrate complex that is there to which an inhibitor has not bound will get converted into product. So catalysis will decrease, the rate of catalysis will decrease, the Vmax will in turn decrease and the Km will remain same. So if you see in the Lineweaver-Burke plot, non-competitive inhibition, the x-intercept remains same because Km is same, whereas the y-intercept changes. So the 1 upon Vmax will be greater than the 1 upon Vmax value for the uninhibited enzyme. Okay. So here you can see the blue line showing the uninhibited enzyme and the red line showing the inhibited version. Okay. Now let's see the last case where the uncompetitive inhibition is happening. So in case of uncompetitive inhibition, you will see there's a change in the enzyme substrate and enzyme substrate binding, that is the Km. There's a change in Km as well as there's a change in Vmax. Why so? If you see here, the initial velocity is reducing. It is reaching the same, almost same Vmax or higher Vmax as in case of non-competitive inhibitor, but the Km and Vmax both has changed from the original uninhibited enzyme. Okay. So if you see in the Lineweaver-Burke plot, you will get parallel lines like this for uninhibited and inhibited enzymes. So the Km has the Vmax has decreased. You can see here this is 1 by Vmax. So 1 by Vmax has increased, that means Vmax has decreased for inhibited, and Km has also decreased in this case. So Km is also Km that is shown on your x-intercept, that is also changing. What is not changing in this case is the slope. The slope, that is the ratio of the Km and Vmax, that is not changing. Okay. So slope for this Lineweaver-Burke plot is basically Km upon Vmax, right? So the slope is not changing. Now why the slope is not changing? If you see the degree of the extent by which Vmax is changing and the extent by which Km is changing is the same. Okay, that value is same. Let's say that value is alpha. Both are changing by alpha. If the Vmax is getting half, the Km is also changing by the same extent. Okay. Okay. So this is clear. So here in this case, A is the competitive inhibitor, B is the non-competitive inhibitor, and C is the uncompetitive inhibitor. This is the mechanism of inhibition in all three cases, and these are the Lineweaver-Burke plot for all three cases. Okay. Okay, moving to the next question, question number 6. Which among the following are true? Now there are 5 statements given. We have to select the true statements. first statement is competitive, non-competitive and uncompetitive inhibitions are all reversible. Second statement is competitive inhibition cannot be overcome by increasing the concentration of the substrate. Third statement is non-competitive inhibition is irreversible, whereas competitive and uncompetitive are reversible. Fourth case is competitive inhibition is irreversible, whereas non-competitive and uncompetitive inhibitions are reversible. Fifth statement is non-competitive and uncompetitive inhibition cannot be overcome by increasing the concentration of the substrate, whereas competitive inhibition can be overcome by increasing the substrate concentration. So select the correct statements amongst these 5 statements. So which of the following statements are correct? Anybody would like to answer? Okay, let's see. First statement, whether the first statement is correct or not. Tell me, competitive, non-competitive and uncompetitive inhibitions are all reversible. Is it true or false? It is false. Neeta is saying it's false. Okay. So if you have seen the mechanism, just a second. If you have seen the mechanism here, if I show you this diagram, the scheme of all 3 inhibitions, now will you say the inhibition is reversible or irreversible? Tell me, all 3. Okay. So Neeta is saying reversible include competitive and non-competitive. So why do you think uncompetitive is not reversible? If you see here, all 3 inhibitions are actually reversible. So if you see, all 3 inhibitors are binding to the enzyme with the equilibrium constant between them. So all 3 of them are reversible. Even this inhibitor can get released from this enzyme substrate complex and it can go towards this reaction. This can also, the inhibitor can get released from the enzyme and the reaction can proceed towards the product. And this is obviously, I think competitive inhibition is very clear why it is reversible. But I can understand there can be a doubt between the other 2 types of inhibition, non-competitive and uncompetitive. So all 3 inhibitions are actually reversible. Okay. So all three types of inhibitions that we have studied, competitive, non-competitive and uncompetitive, all 3 inhibitions are reversible, because in all 3 cases the inhibitor can actually detach from the enzyme. The binding is not covalent kind of binding where the breakage can hamper the activity of the enzyme. Okay, so the inhibitor can dissociate from the enzyme and then enzyme can bind to the substrate and product formation can happen. So that can always happen in all 3 cases. Okay, and so the second okay, so the third and fourth statement could not be true because we have discussed all of them are reversible. Amongst the second and fifth statement, the fifth statement is the true one. Why the fifth statement is true? Because if we go to the scheme, so I've told you there's a competition between the substrate and inhibitor. So if we keep on increasing the substrate concentration in case of competitive inhibition, there will be a point where the Vmax will be achieved. So substrate increasing the substrate concentration can actually uh, help in achieving the same Vmax. So the substrate, more and more substrate is added to the enzyme, more and more substrate can bind to the enzyme. It can compete uh, with the inhibitor for the binding site. Okay. Whereas in the other two cases, that won't be the case, because it's not the substrate and inhibitor that is fighting for the same site. Okay. Inhibitor, even in presence of a lot of substrate, there will be inhibitor that can sit in some other site, because there is no competition for the inhibitor site. The inhibitor site is different from the active site of the enzyme. Okay. But in case of competitive inhibition, the same active site is there for both inhibitor and substrate. So in that case, actually increasing the substrate concentration can be used to overcome the competitive inhibition. Okay. So competitive inhibition can be overcome by increasing the concentration of substrate. So option B, that is first and the fifth statement, is correct. I hope this is clear. Neeta, is it clear why all three enzymes are actually reversible? All three inhibitors are actually reversible inhibitors? Okay, very good. So now we'll move forward to the next question. That is question number 7. Salivary amylase is an enzyme in humans that breaks down starch. The optimum pH for this reaction is 6.7. It is also known to be associated with calcium ion Ca2+. In conditions lacking calcium, the enzyme would be referred to as A. The rate of this enzyme reaction would not be affected by the condition depicted as B. A and B are respectively dash. Okay, so you have to identify A and B. So A is where the enzyme is not associated with calcium. In absence of calcium, what would you call the enzyme to be? And B is the condition in which the enzyme's activity won't be affected. Okay, so now we have the option. Option A says A, that is the enzyme, would be referred to as coenzyme, and the rate of this reaction would not be affected by maintaining the pH of reaction at more than 6.7. Okay. The second option is A is apoenzyme, and B, that is the condition, is maintaining the pH of the reaction at 6.7. C is again coenzyme and maintaining the pH of the reaction at 6.7. And D says the enzyme would be referred to as apoenzyme, and the condition should be maintaining the pH of the reaction more than 6.7. So the optimum reaction in the question, optimum pH in the question is given as 6.7. So what should be the answer? Anybody, what should be the answer? This is very straightforward. Okay, Neeta has answered option C. Anybody else wants to answer? So any enzyme, if it is bound to a small molecule, that enzyme, like if it is active only in presence of the small molecule. So we talked about the allosteric control, right? So this is kind of like that, but the term that we use to refer to the enzyme without that small molecule. If the small molecule is not there, then that enzyme will not be fully active. It's an incomplete enzyme, you can say, and that enzyme is called the apoenzyme. Okay, and the condition should be maintaining the pH of the reaction at 6.7. So that is pretty straightforward. So if your optimum pH for a enzymatic reaction is given, that has to be the pH for the reaction. Okay, so if you are carrying out some enzymatic reaction, you have to maintain the pH of your reaction system according to the optimum pH of the enzyme. Okay, so any enzymatic reaction follows a pH curve, that is a bell shaped curve. So I can draw it roughly. So if I have a pH scale here and the enzyme activity here, so you will find a bell shaped curve. So this the point where the maximum activity is seen is called the optimum pH of that enzyme. Below and higher than the optimum pH, you will get a lower activity. Okay, so if I go below this pH, optimum pH, or if I go above this optimum pH, in both cases you will find the activity is lesser than what it was at the optimum pH. Okay, so always the reaction condition has to be maintained at the optimum pH. So the correct answer is option B, that is the enzyme is called apoenzyme and the pH has to be maintained at the optimum pH. So here's the diagrammatic representation of apoenzyme. So if you see, without the cofactor, the enzyme is called the apoenzyme. Once the cofactor binds to it, it is called holoenzyme. So without the cofactor, the substrate cannot bind to the enzyme. So the difference between the allostery and this concept is that in case of allosteric effect, the cofactor or the small molecule that was binding, it was not binding at the active site. Okay, and in case of allostery, there was a regulation. So it can either enhance the activity of the enzyme or it can inhibit the enzyme, reduce the activity of the enzyme. But in case of cofactor, that cofactor is actually necessary for the enzyme catalysis. Without that cofactor, the enzyme will always be inactive and that cofactor will be found in the active site of the enzyme. So it's generally a metal ion that are used as cofactor, or some FAD or NADH molecules. These are all small molecules that are used as cofactors in an enzyme. So let's say DNA polymerase. So polymerase has magnesium ions that are used as cofactors. Okay, so when that metal ion will sit in the active site, only then the substrate will be able to fit in the active site and the reaction will be catalyzed with the help of the metal ion. Okay, so without the cofactor, the enzyme is called the apoenzyme. Once you have both the cofactor in the enzymes, like cofactor and the enzyme together, it is called the holoenzyme. So this is the active form of the enzyme. Apoenzyme is the inactive form of the enzyme. It is fully folded properly in proper conformation, but still it is inactive because the cofactor is missing. So it is the apoenzyme. Once the cofactor is there, it is called the holoenzyme. So you can remember, like, it's whole. Okay, so holoenzyme, it's complete in nature, it's whole in nature. Okay, whole as in WHOLE. Okay, so that way you can remember. So holoenzyme and apoenzyme. This is clear? But apoenzyme does not bound while coenzyme as a protein do bound. Neeta, I did not get your doubt. Can you be more specific? So if I understand what you are saying that apoenzyme will not bind to the substrate, right? So coenzyme is the cofactor that is binding to the enzyme. So this blue part that is shown here, apoenzyme not necessary for enzyme activity. So no, so I think you are not getting the point here. Wait, I'll explain. So just a second, so if I have an enzyme, okay, so this enzyme needs a helper. Okay, this enzyme needs a helper that is called coenzyme or cofactor. Okay, so coenzyme or cofactor, it's a small molecule. Small molecule, generally a metal ion. Metal ion is one example. Okay, so this small molecule, once it binds to that enzyme, now this enzyme is completely functional. Okay, so this holoenzyme that I am saying, this is the active form of enzyme. So holoenzyme is nothing but the complete enzyme, okay, that now that substrate can bind to the enzyme. If I remove this coenzyme or cofactor from this enzyme, so now I have the enzyme, okay, and I have removed the cofactor from it. So once the cofactor is removed, the remaining part of the protein, so enzyme is nothing but a protein, right? Folded protein with a catalytic pocket which can catalyze a particular reaction, right? That is the enzyme. So once the cofactor is removed from that enzyme, the remaining protein, the remaining folded protein with the catalytic site, that is called apoenzyme. Okay, that is called apoenzyme. So if you see in this question, it was said that some salivary amylase is known to be associated with Ca2 plus. So this amylase enzyme, it is known to be associated with calcium. Now amylase plus calcium is the active form of enzyme. Okay, amylase plus Ca2 plus, it is active. Okay, if I remove this calcium, so amylase minus Ca2 plus. Okay, if there is no amylase, if there is no Ca2 plus with the amylase, this amylase will be called as apoenzyme. Sorry, is it clear now? I hope this is clear now. So apoenzyme, yeah, okay. So apoenzyme means just if you remove the cofactor from the protein, the remaining part of the protein, the remaining part of the enzyme is called the apoenzyme. You add the cofactor again, and again the enzyme will be completely active, and it is called the holoenzyme. Okay, so two parts. This cofactor that is binding to the enzyme and getting released, this basically in this case calcium is called the coenzyme or the cofactor. Generally we use the term cofactor because most of the time these are small molecules, so these are not any other enzyme. So in some cases it could be possible that one enzyme needs another enzyme for its activity, so in that case coenzyme is the better term. Okay, so generally the term cofactor is used, and most of the time the enzymes have a metal center, metal ion center in its active site. So most of the cofactors are actually metal ions. So it could be calcium, magnesium, iron containing. There are so many iron containing proteins. Then you could have copper containing proteins. So like that. Okay, so these are called the cofactors, and when they bind to the enzyme, it makes the enzyme completely active. So this is clear. Okay, moving to the next question. So question 8 and 9. The given diagram shows a certain inhibitory effect where E stands for the enzyme, S stands for the substrate, and I stands for the inhibitor. For the depicted enzyme substrate interaction shown, the inhibitor I can bind either to enzyme or enzyme substrate complex. So, E is the enzyme, S is the substrate, I is the inhibitor. So, here they have shown that the inhibitor can bind either to the free enzyme or it can bind to the enzyme substrate complex. So, I have already shown you such diagram, so you should be able to answer this question. So, based on this diagram, we will have two questions here. Question number 8 is identify the inhibitory process shown. Uncompetitive, competitive, non-competitive, or the reaction is not inhibition reaction. So, it is an inhibition reaction, you know that since there is an inhibitor. So, tell me what type of inhibition is happening. I have already discussed what type of inhibition is happening here. Come on, answer fast. We have already discussed this, so you should be able to answer. I need an answer from you, from your side. The inhibitor can bind to both the free enzyme and the enzyme substrate complex. It's not competitive. Try again. So, if you remember, so this type of inhibition is called non-competitive inhibition. So, if you remember here I had shown three diagrams, right? So, if the inhibitor is binding only to the free enzyme, either the substrate can bind to the enzyme or the inhibitor can bind to the enzyme. In that case, it is competitive, okay? So, only inhibitor is binding only to the free enzyme. In the second case, in case of non-competitive inhibition, what is happening? The inhibitor can bind to the free enzyme as well as inhibitor can bind to the enzyme substrate complex, okay? So, inhibitor can either bind to the free enzyme or the inhibitor can bind to the enzyme substrate complex. In the third type of inhibition, that is uncompetitive inhibition, what is happening? The inhibitor can only bind to the enzyme substrate complex. Before binding of the substrate, the inhibitor cannot bind to the free enzyme, okay? So, here if you see what is happening, the inhibitor can bind to both the free enzyme and the enzyme substrate complex. So, it is a type of non-competitive inhibitor. Here in the question you can see it is written the inhibitor I can bind either to the enzyme or the enzyme substrate complex, okay? So, it is called the non-competitive inhibition, okay? This is clear? So, this is the most important part about enzyme inhibition. So, if you have inhibition kinetics, you will have these three types of inhibitions, majorly these three types of inhibitions and their kinetics, questions related to their mechanism of inhibition and their kinetics, okay? So, you have to pay attention to this very carefully. In case of competitive inhibition, since there is only one side. First remember, in case of competitive inhibition, there is only one side, the active site of the enzyme. To that site only, both the substrate and inhibitor can bind. So, at one point, at any point, inhibitor can only bind to the enzyme when substrate is not bound. Okay. So, that means inhibitor can only bind to the free enzyme. In the second case, what is happening? We'll open the diagram again, it would be easier. Yeah. So, in first case, in case of competitive inhibition, inhibitor can only bind to the free enzyme. In the second case, what is happening? Inhibitor is not binding at the active site, it is binding somewhere closer to the active site. So, inhibitor can bind to the free enzyme, as well as it can bind to the enzyme even after the substrate has bound. So, there is an enzyme and I have added the inhibitor and the substrate in the reaction mix. Okay. So, now substrate can bind to the enzyme even after the substrate has bound to the enzyme, the inhibitor can bind to that enzyme. Okay, and also the inhibitor can bind to the free enzyme. So anything can happen. So this is non-competitive inhibition. Uncompetitive inhibition says that it requires the substrate to bind to the enzyme first, only then the inhibitor can bind. Inhibitor cannot bind to the free enzyme. Inhibitor will bind only and only after the substrate has bound to the enzyme. Okay. So this is clear. So this is a non-competitive inhibition. Okay. So the next question, based on the same diagram, the depicted enzyme substrate mechanism has a structural specificity. Identify the species unique to this kind of inhibition. So there is a specific structure that is forming in this kind of inhibition. So what is it called? Option A, the ternary complex formation. Option B, formation of a binary complex. Option C, formation of a quaternary complex. And option D, formation of a polar intermediate. Okay. So what is happening? Ternary complex formation, binary complex formation, quaternary complex formation, or a polar intermediate formation? What is happening here? Yes, ternary complex formation. Very good. So if you see, what do you mean by ternary complex? It means basically 3 different units combining together. Binary means 2 units. Quaternary means 4 units. Okay. So here a special ternary complex formation happens. This is not there in case of competitive inhibition. This is only there in case of non-competitive and uncompetitive inhibition. So the ESI, the complex of enzyme substrate inhibitor, 3 units are there, enzyme is there, substrate is there, inhibitor is there. So 3 molecules are there. So all 3 molecules coming together and forming a complex, it is called a ternary complex. Okay. So ternary complex formation is happening. Sorry. Yeah. The correct answer is option A, ternary complex formation. I hope this is clear. The concept of inhibition, competitive, uncompetitive and non-competitive. Tell me if that is clear. Otherwise, I'll explain it again, because it is very important for you guys to understand the concept very clearly, the difference between all 3 types of inhibition. Okay. I hope everyone has understood, because only one person has responded. If there is any doubt, either you can unmute or you can write in the chat box if you have any kind of doubt. Okay. Okay. Moving to the next question. Next question is, RNase uses one of the following mechanism of catalysis. Option A, covalent catalysis. Option B, metal ion catalysis. Option C, acid base catalysis. And option D, all of the above. RNase uses one of the following mechanism of catalysis. Okay, RNase is an enzyme. It's also called ribonuclease. So it uses which of the following mechanism? Anybody? Covalent catalysis, metal ion catalysis, acid base catalysis, or all of the above? So the answer is option C, acid base catalysis. Okay, so ribonuclease, what it does, it cleaves the RNA molecules. Okay, RNA is ribonucleic acid. So it is a chain of ribose sugar bound to different bases. Okay, and the binding is through a phosphate linkages, phosphodiester linkage, okay, and that phosphate linkage is cleaved by this ribonuclease or RNase enzyme, okay. So we'll discuss the mechanism shortly. Let's see the next question first. Question number 11. The catalytic reactive group in the active site of RNase are. So there are two catalytic active sites in the RNase enzyme. So I told you that the RNase cleaves the ribonucleic acid, the RNA molecule. It chops off the RNA molecule between their phosphodiester linkages, okay. And in order to do that cleavage, two amino acid residues that are present in the active site of RNase, they facilitate this cleaving reaction, that is the acid base catalysis. So what are those two amino acid residues? The two amino acid residues are actually histidine 12 and histidine 119. Now we will see how. So this is the complete mechanism of ribonuclease. So here you can see this is the RNA molecule. So you have the ribose sugar, okay, ribose sugar, where there is a OH at 2 prime and OH at 3 prime, okay. Ribose sugar, at the first carbon the base is added. So base here does not mean the acid or the base. The base is basically the nucleic acid, the bases of the nucleic acid. So basically adenine, guanine, cytosine and thymidine. So among those four molecules, one of the molecule will be here, and then from the third carbon there is a phosphate group that forms the linkage between the third carbon of the first ribose and the second carbon of the ribose, okay. Okay, so this is 1 2 3 4 fifth carbon of the ribose, okay. This is clear. So this is the main structure of RNA. We will discuss the structure of nucleic acid in the upcoming lectures. So at that time you will get the better idea of how a RNA looks like. But for now, for an idea, this is pretty nice. So you have a ribose sugar with two ribose sugars and bases. These nucleotides are attached together with a phosphodiester linkage. And what ribonuclease, the enzyme, does, so this is the enzyme's catalytic pocket. So this is the enzyme's catalytic pocket, and you have the two histidine residues in the enzyme's catalytic pocket, which basically catalyzes the reaction where the breaking of this ribose molecules joined together by this phosphodiester bond, this happens, okay. So this ribonuclease enzyme, RNase, it breaks down the RNA molecule at the phosphate linkage, okay. We will see how. So now I have told you there are two amino acids at the catalytic site, histidine 12 and histidine 119, and I have told you the type of catalysis that is happening is acid base catalysis. So what is happening is, first this histidine 12 acts as a base and abstracts the proton from the second 2 prime OH group, okay. So here, for a clear explanation, I will draw it here. So this is your ribose sugar, right? And so we will have the base here, and then the OH group is here, and then another O, and then PO4 is there. So the rest of the structure, and here CH2, okay. So this hydrogen that is there, this proton, it is abstracted by the histidine 12 residue in the active site. So this proton is abstracted. Abstracted means The histidine 12, since it is acting as a base, it can take the proton. It is acting as a proton acceptor. So histidine 12 is acting as a proton acceptor. Okay, proton acceptor or a base. Okay, so this histidine 12, since it is acting as a base, it will accept a proton from the second prime OH. Once this proton is taken out, what will be left here? The O minus will be left here. Okay, so if I remove this proton from here, just a second. So now if I remove this proton from here, what will happen? O minus will be there. This negative, this O minus will now attack this phosphate group. Okay, this O minus can now attack this phosphate group, and what will happen, since O minus is attacking this phosphate group, and we have another histidine residue, histidine 119, which is acting as an acid. Now histidine 12 was acting as a base, so it was accepting a proton. Histidine 119 is acting as a acid, so it will donate the proton. So proton donor. Okay, so it will donate the proton. So what is happening? So this proton is abstracted by histidine 12. After the proton abstraction, oxygen is there with a negative charge, so O minus. This O minus will attack the phosphate. Once the O minus attacks the phosphate, this histidine 119 will donate its proton to this oxygen, and the bond will get cleaved here. Okay, and a cyclization reaction will happen. Since this oxygen has attacked this phosphate, so oxygen will bound to the phosphate and a cyclic intermediate will form, and this particular base, the second base that is there, it will be released with the hydrogen attached to it, that has come from histidine 119. So this hydrogen has come from histidine 119. Okay, and this histidine 12, this is histidine 12. Histidine 12 has the hydrogen that was there with the 2' OH group. Okay, now for an enzyme to be active again, it has to revert back to its original form. Okay, so whenever there is an enzymatic reaction happening, once the enzyme has done its reaction, it has to go back to its original form so that another molecule of substrate can bind to the enzyme and the catalysis can be continuous. Okay, so it's a catalytic cycle happening. Okay, so enzyme is kind of it can be used and reused again and again. Okay, so here you see histidine 12, now it has to again donate this proton that it had in order to go back to its natural state, where it can again abstract a proton from the next substrate. Okay, so now if it is donating a proton, now what is happening? The histidine 12 is actually acting as an acid. Okay, and histidine 119 had donated its proton to the cleaved molecule. So now histidine 119 has to accept a proton in order to go back to its natural side. So histidine 119 acts as a base. Okay, so what was happening in first case? Histidine 12 was proton acceptor and histidine 119 was proton donor. Once the cyclic intermediate has formed, in the second step what is happening? Histidine 12 now will act as proton donor because it already has the proton that it has accepted. So now it will act as the proton donor, or what we call as acid, and histidine 119 will act as a proton acceptor, or what we call it as base. Okay, so in first case histidine 12 was the base and histidine 119 was the acid. Now the roles has reversed. Now histidine 12 is the acid and histidine 119 is the base. So we have histidine 12 and histidine 119. Now what will happen, there will be a water molecule from which histidine 119 will abstract the proton. So it will act as a proton acceptor. Once this proton has been abstracted, what we will get? We will get an OH minus. From H2O, we have H2O, that is the water molecule. If histidine 119 has taken the proton, accepted the proton, what we will get? OH negative. OH negative can actually attack this phosphate. Like how here O negative was attacking this phosphate, now OH negative will attack this phosphate. Once OH negative attacks this phosphate, this bond will again break and this oxygen will accept the proton that is there with the histidine 12. So the proton that histidine 12 had taken in the first case, that proton will be given back to the oxygen in the second step. Okay, so the molecule will revert back to its original form and there will be a bond breakage. Okay, so this is how ribonuclease, or we call it in short RNase, acts. Okay, so RNase basically breaks down the RNA molecule into its individual nucleotides. Okay, and this is the mechanism of how it is working. And the two main amino acids that are there in its active sites are histidine 12 and histidine 119. And the type of catalysis is acid base catalysis. Why acid base catalysis? As you can see, there's a one residue is acting as acid and second residue is acting as base in first step, and the roles are reversed in the second step. Okay, so this is simple acid base catalysis example. Okay, I hope this mechanism is clear because this is very important. Okay, so for the next two questions, go through this mechanism first. Okay, from the given diagram, we have to answer the question number 12 and question number 13. So question number 12 is, the diagram shows the working of the enzyme dash by the dash mechanism. So which enzymes mechanism is shown here? Options are lysozyme and ribonuclease. So I have already shown you the mechanism of ribonuclease, so you should be able to answer this. This is the mechanism of lysozyme. Okay, and what is the mechanism called? The mechanism is called the Phillips mechanism. Okay, so here the mechanism of the enzyme lysozyme is shown, and the mechanism is called Phillips mechanism after the scientist's name who has discovered this mechanism, or who has given this mechanism. Okay, who has described this mechanism. Fine. So lysozyme's mechanism is given. So option number A is the correct answer. The enzyme lysozyme and the mechanism is Phillips. So what is happening in this enzyme? We will see the mechanism of lysozyme once. So lysozyme, what it does, it breaks down the bacterial cell wall. Okay, bacterial cell wall is mainly made up of a polymer called peptidoglycan. Okay, and that peptidoglycan is nothing but an alternating structure, like alternating units of two sugars, NAM and NAG. Okay, so N-acetyl glucosamine and N-acetyl muraminic acid are two residues that are there in the peptidoglycan in an alternating manner. Okay, so this is a NAG molecule NAG, and this is a NAM molecule NAM. So N-acetyl glucosamine and N-acetyl muraminic acid. Okay, so such repeating unit, it's a polymer, so it's a long chain of NAG, NAM, NAG, NAM, and so on. I'm sorry. Yeah. So from that polymer, in the lysozyme enzyme's active site, so whatever the active site of the enzyme is there, 6 of those units sit in the lysozyme site. Okay, so lysozyme is the enzyme, it has the active site, the catalytic pocket. In that catalytic pocket, 6 units of NAM and NAG sits together. Okay, so here you can see 1, 2, 3, 4, 5, 6, or it is labeled as A, B, C, D, E and F. Okay, so 6 units. So NAG, NAM, NAG, NAM, NAG and NAM. Okay, and from these 6 units, once these 6 units fit in the catalytic pocket, the cleavage occurs always at the after the 4th residue. So A, B, C, D, the cleavage will happen between D and E residue at the carbon oxygen bond of the glycosidic linkage. Okay, so here the zoomed version is shown, so you can see the carbon 1 and carbon 4 is attached by a glycosidic linkage, and always the bond between the first carbon and the oxygen will break between the D and E residues. Okay, so this will all, the enzyme is so specific towards its substrate that it will always and always do the cleavage at that particular site. Okay, so that is the beauty of the enzyme. Now let's look at the mechanism. So what is happening, like in case of ribonuclease, I've shown you there were 2 histidine residues in its active site that were doing the catalysis. Okay, so that was an acid base catalysis. Here what is happening, here also something similar is happening. You see there are 2 active residues, glutamic acid and aspartic acid. Okay, so glutamic acid and aspartic acid, both are acidic groups. Okay, both have acidic side chain, right? So here you can see glutamic acid has the COH and the aspartic acid has the CO minus. Okay, so in the first step what happens, glutamic acid actually abstracts this donates this proton to this oxygen and the unit is broken. But once this bond is broken between the oxygen and the carbon, what is happening, an oxonium ion formation is happening. Okay, this is called an oxonium ion formation, where the chair conformation is actually distorted. Once this is formed, so this is basically a positive sign, right? O positive is there, right? So this O positive is stabilized by the negative charge of the aspartic acid. So this is a charge stabilization. So there is no bond formation between the negative oxygen and positive oxygen, but there is a charge stabilization. So non-bonded charge stabilization is there. Okay, so the distances are not very close enough so that the bond formation can happen, but it can actually stabilize the charge. Once this aspartic acid is stabilizing the charge, again this glutamate 35 residue, now it has donated its proton to the unit, the cleaved unit. So now it has the O negative. It can actually, that O negative can abstract a proton from a water molecule and resulting in a formation of OH negative that can act at the oxonium complex, and the OH can bind to the remaining part of the chain. Okay, so here what is happening, this bond is breaking and at the fourth carbon CO, one H is coming, and at the first carbon also CO, one H is coming. So both OH, both groups are getting OH at the end, right? So mechanism is clear. So this is the mechanism for lysozyme. Now moving to the next question, question number 13. The enzyme whose mechanism is shown is specific for catalyzing at which bond? Okay, so NAM-NAG glycosidic bond of the beta-1,4 conformation, NAM-NAG glycosidic bond of the beta-1,2 conformation, NAM-NAG glycosidic bond of the alpha-1,4 conformation, and NAM-NAG glycosidic bond of the alpha-1,2 conformation. So I have already told you it's a glycosidic bond. So is it a beta-1,4, beta-1,2, alpha-1,4 or alpha-1,2 conformation? So the answer is beta-1,4 conformation. Okay, so option A. Why beta-1,4? So here if you see the linkage, so the linkage is basically between the first carbon of the NAM residue and the fourth carbon of the NAG residue. Okay, so first and fourth, the linkage is 1,4. And why it is beta? Beta because the OH of both the groups are in opposite direction. So they are trans to each other. So one is coming out of the plane and one is going inside the plane. So if both the OH are going inside the plane and making a bond, it will be called an alpha glycosidic linkage. In other case, it will be called beta glycosidic linkage. So this is a beta glycosidic linkage between the first carbon of NAM and the fourth carbon of NAG. So this is called beta-1,4 linkage. Okay, this is clear. Okay, so the upcoming questions are based on this Lineweaver-Burke plot. So we have already discussed this Lineweaver-Burke plot, so I hope this should not be a problem. So question number 14 is state which graph represents which process. Option A is competitive, uncompetitive and non-competitive inhibition. Option B is non-competitive, uncompetitive and competitive inhibition. Option C is competitive, non-competitive, uncompetitive inhibition, and option D is uncompetitive, non-competitive and competitive inhibition. So let's see. So A, B and C, three type of inhibitions are shown. So in case of A, both Vmax and Km is changing. In case of B, Km is constant and Vmax is changing. And in case of C, Vmax is constant and Km is changing. So which is what? So as I have told you, in case of competitive inhibition, since the inhibitor and the substrate is competing for the same site, if we keep on increasing the substrate concentration, the enzyme can reach that same Vmax. Okay, so the Vmax does not change, but the Km increases. So in case of competitive inhibition, the Vmax does not change. So in the other two cases, the Vmax is changing. So C is competitive inhibition. Now in case of non-competitive inhibition, we have the Km constant and Vmax decreases. Okay, the Km does not change because the substrate binding does not get affected. The inhibitor can bind to both the free enzyme and the substrate. So substrate binding is not hampered. Thus the Km is constant, only the Vmax is decreased. So non-competitive inhibitor is shown by the graph B. And if you see the graph A, where both Vmax and Km is changing, that is the uncompetitive inhibitor. So the answer is uncompetitive, non-competitive and then competitive. So option D is the correct answer. I hope this is clear. So option D is the correct answer for this case. Now, based on the type of inhibition occurring Identify the correct changes in the Michaelis-Menten constant and the Vmax in the process. So we have already discussed this again. So in case of uncompetitive inhibitor, so let's see, in case of uncompetitive inhibitor, we can see the Vmax is decreasing. The 1 by Vmax is increasing. That means Vmax is decreasing. So A, Vmax is decreasing. Okay. And Km is also decreasing. So Km is also decreasing, right? In case of B, Km is constant and Vmax is decreasing. So Km is constant and Vmax decreases. And in case of C, Vmax is constant and Km is increasing. Okay. So you see here, Km is increasing. Minus 1 by Km is going towards the left hand side, that right hand side. That means Km is increasing. Whereas in case of uncompetitive inhibitor, the Km is decreasing. Okay. So that means option A is the correct answer. Km decreases. Km and Vmax both decreases in case of uncompetitive inhibition. Km is constant and Vmax decreases in case of non-competitive inhibition, and Km increases and Vmax is constant in case of competitive inhibition. Okay. So this is clear. Question number 16. The turnover number is defined as the maximum number of molecules of substrate that the enzyme can convert product per catalytic site per unit time. Okay. So in the last class also we had discussed Kcat. So it is the maximum number of molecules of the substrate that the enzyme can convert into product per catalytic site per unit time. It is calculated as Kcat is equal to Vmax upon Et. Identify the correct ways to identify the correct ways in which the inhibition process shown in the figure affect the turnover number. Now we have to tell how will the turnover number or the Kcat will be affected in each case of inhibition. So Kcat is given by Vmax by Et. So Kcat is depending on the Vmax. Kcat is directly proportional to Vmax. Okay. So let's see here, or here, option A is the correct answer, right? So if Kcat is directly proportional to Vmax, then if Vmax is decreasing, Kcat will decrease in case of uncompetitive inhibition, and in case of non-competitive inhibition also Kcat should decrease because Vmax is decreasing. But in case of competitive inhibition, since Vmax is constant, Vmax is not changing, Kcat will also not change. Okay. So answer is option B. There will be a decrease in case of uncompetitive and non-competitive inhibition, but there will be no change, or the Kcat will be constant in case of competitive inhibition. Okay. So this was question number 16. Any doubt in these three questions? These three questions were based on the Lineweaver-Burke plot of three types of inhibitions that we had discussed earlier also. So main thing to remember is, in case of uncompetitive inhibition, both Vmax and Km are decreasing. In case of non-competitive inhibition, Km is constant but Vmax is decreasing. And in case of competitive inhibition, there is no change in the Vmax and the Km increases. Okay. Neeta has requested to repeat 13 and 14 question. Yeah, sure, I can repeat that. So 13 and 14. So 13th question was the enzyme whose mechanism was shown. So in that case, lysozyme's mechanism was shown, right? So which bond is catalyzed by lysozyme? That was the question. So the answer is beta 1,4 linkage. So I had already explained, I'll explain again. So here this is the bond between the NAM and NAG unit. Okay, so the bond is between the first carbon of the NAM and the fourth carbon of the NAG, and this bond is called the glycosidic bond. Okay, glycosidic bond. And so the glycosidic bond is formed between the first carbon of NAM and fourth carbon of NAG. So this is basically a 1,4 linkage, 1,4 glycosidic bond. Now how to decide whether it is alpha or beta? So in case of beta glycosidic bond, the OH groups will be opposite to each other in terms of their in terms of their geometry. So if this OH is coming out of the plane, this will make beta 1,4 linkage. If this OH is also going inside the plane, both the OHs are towards the same direction, it will make a alpha 1,4 linkage. Okay. So this was the question. The enzyme whose mechanism is shown is specifying for catalyzing the NAM-NAG glycosidic bond of beta 1,4 conformation. Okay. And question number 14 to 16 were based on this diagram. So this diagram I have already shown. So the question number 14 was, what is the type of inhibition? So you had to identify what is the type of inhibition in each case. So I had already gone through this before. So this is the, so in this case the Vmax is changing and the Km is changing. So this is called uncompetitive inhibition. In this case, in the second case, the Km is constant but Vmax is decreasing. It is called non-competitive inhibition. And in the third case, C, the Vmax is constant and the Km is increasing. It is called competitive inhibition. Okay. So option D is the correct answer. Okay. So that was the question 14, and then based on that only, what is the changes in Km and Vmax, question number 15, and then what is the effect on catalytic turnover number, Kcat, catalytic efficiency basically. So Kcat is defined as Vmax upon ET. ET is basically the total enzyme concentration. So Kcat is directly proportional to Vmax. So if Vmax is decreasing, Kcat will decrease. If Vmax is increasing, Kcat will increase. If Vmax is constant, Kcat will be constant. Okay. Okay. So moving to the next question, question number 17. Which one among them is the example of competitive inhibition of an enzyme? Okay. So succinic dehydrogenase by malonic acid, cytochrome oxidase by cyanide, hexokinase by glucose 6-phosphate, and carbonic anhydrase by carbon dioxide. So which of the following is an example of competitive inhibition? Anybody knows the answer? Which of the following is an example of competitive inhibition? Succinic dehydrogenase by malonic acid, cytochrome oxidase by cyanide, hexokinase by glucose 6-phosphate, and carbonic anhydrase by carbon dioxide. Okay, option A. Yeah, very good. So option A is the correct answer. So here you can see succinic dehydrogenase, what it does, it converts succinate into fumarate. So this is the structure of succinate, and what it does, it basically introduces a double bond between these two carbon to convert it into fumarate. Okay. Malonate, if you see the structure of malonate and succinate, they are very similar. Only one extra CH2 is there in case of succinate. Okay. So malonate can actually fit in the catalytic pocket of succinate dehydrogenase. But since the reaction is very specific, as I've mentioned that enzymes are very specific towards their substrate. So if another molecule that is mimicking the structure of its substrate fits into the catalytic pocket, in that case also there won't be any catalytic activity or catalytic reaction that will go ahead or go forward. Okay. So malonate is called a competitive inhibitor because it sits in the same pocket as the succinate, as the substrate. So inhibitor and substrate binds to the same active site. Okay, but malonate does not get converted into any product. It inhibits the enzyme. It basically prevents the succinate from binding. So both succinate and malonate will compete for the same site. So it's a competitive inhibitor. Okay, it's also called a substrate mimic. Sometimes we call the competitive inhibitors as substrate mimics because they mimic the structure of the substrate and they fool the enzyme by making it think that it is the substrate, and the enzyme binds to that substrate mimic instead of the substrate. Okay. Okay. So, question number 18. In a competitive inhibition, what happens to Vmax and Km if the concentration of I is equal to Ki? Okay, so this is the last question. In a competitive inhibition, what happens to the Vmax and Km if I is equal to the Ki? So, what will happen to the Vmax and Km? What will happen to the Vmax in case of competitive inhibition? We have discussed this for so many times. First, tell me what will happen to the Vmax. Come on. What will happen to the Vmax in case of competitive inhibition? Is it changing? So in case of competitive inhibition, Vmax doesn't change. I've explained it again and again. So Vmax will not change. So Vmax is unchanged. And what will happen to the Km? Let's see. So in any competitive inhibition kinetics, so the correct answer is option B, obviously. But what happens is, in the competitive inhibition kinetics, the kinetic reaction, the Michaelis, you know, the Michaelis-Menten reaction for any uninhibited enzyme is V is equal to Vmax into the substrate concentration divided by the Km plus the substrate concentration, right? So I am saying that Vmax doesn't change in competitive inhibition and Km increases. So Km will increase by a factor of alpha. So this is for inhibited. This is for inhibited enzyme. Okay. And Km is increased by a factor of alpha, that is written as 1 plus I by Ki. Okay. Now I am saying in the question, I is equal to Ki, right? In the question it is given I is equal to Ki. So if I is equal to Ki, what do we have? We have this will be equal to Ki. So 1 upon 1 will be 1 and 1 plus 1 is 2. So if I is equal to Ki, alpha will be equal to 2. So that means the new Km is what? 2 times the actual Km. Okay. The new Km, the inhibited Km, is actually increases by 2 times. Okay. The Km is increasing 2 times. Okay. So Vmax won't change and Km will increase. Km will get double. Okay. 17. Okay. So this is the question 17 slide, Neeta. And do you have any doubt in question 17? Okay. Okay, so that was the last question of this session. So anybody has any doubt in any of the question, any of the 18 questions, or if you have any doubt in general in this topic, you can ask me. If we don't have any doubt, we can end this section. Okay. I hope everything is clear. If if you have any doubt, you can either ask right now, or you can also I think post your doubts in the portal also. So just go through it, go through all the lecture videos that are posted by Professor Swagata, and if you have any doubt how to solve the questions, you can see the problem solving session videos also on the same portal. The links will be shared. So this whole session has been recorded, and this will be uploaded on YouTube, and the links will be shared with you. So feel free to go through the videos whenever you want to, and I'll see you next week again. So I'll stop the session now. Thank you everyone for joining.